Welcome to the KEMRI-Wellcome Trust Research Program, Biosciences Department. My name is Isabella Oyer. I joined the program 14 years ago as a postdoctoral researcher, having completed my PhD at the Liverpool School of Tropical Medicine. I have over the years been supported to obtain internationally competitive research grants to develop into an independent scientist focusing on malaria molecular epidemiology. I am now the head of the Biosciences Department. Within the department, we focus on 5 main research areas: malaria, viral and bacterial diseases, neglected tropical diseases, and maternal and child health. And what we do in this area is understand why people get sick with these diseases. We want to understand how their body is fighting off infection, looking at antibodies so that we can inform vaccine design. We also want to understand the pathogen or the organism that's making them sick. And one of the ways we do this is by looking at the genetic material of this pathogen to understand how it's moving in populations and how it's transmitting from one individual to the next. We are involved in vaccine trials and clinical trials. So what different drugs can we use to cure some of these diseases? All this research is supported by a well-equipped GCLP accredited laboratory platform. And it's because of all these years, several years of investment in infrastructure and human resource that we've been able to now conduct over 30,000 COVID-19 tests. So I will now be happy to take you through the lab to show you the different areas where we're working to support the COVID-19 testing. This is our microbiology laboratory where we routinely conduct diagnostic testing for the Kilifi County Hospital to understand microbiological organisms, and we support research in bacterial diseases and viral diseases. We are now using this laboratory facility as a dedicated area for COVID-19 testing. And the reason we're able to do this is because we have a number of Category 2 hoods that allow us to safely receive the COVID-19 samples from 6 coastal counties and to process them in the Category 2 hood, as you see behind me with my colleague who is now processing some samples we received today from the 6 coastal counties. One of our assets within the Biosciences Department is our biobank facility. Within this facility, we have over 1 million samples stored in -80 freezers. And these are patient samples that come from the community or from hospital facilities. These samples are blood, urine, and stool, just to name a few. So we've also, again, exploited this facility to support the COVID-19 testing, where we have now a dedicated freezer for the COVID-19 samples that we've been receiving from the 6 coastal counties. In addition to our molecular COVID-19 diagnostic testing, we have exploited our knowledge in proteins for the various diseases we work on: malaria, chikungunya, influenza, pneumonia, to inform how we can design proteins against the SARS-CoV-2 virus so that we can use this protein to understand how many individuals in the population have been exposed to the SARS-CoV-2 virus. And we do this within this facility that we call the cell culture room. Once our colleagues have finished processing the samples in the microbiology laboratory, they then move over to the immunology laboratory where we have the molecular facilities. It is within this room that we are in now, what we call the PCR master mix preparation room, that they mix the reagents that will be used for the qPCR test. We're now at the final section of our tour, which is the molecular biology area within the immunology laboratory. It's in this area that is dedicated space for molecular work, where we look at DNA and RNA. And these machines over here, the real-time PCR machines, allow us to conduct the real-time PCR molecular diagnostic test for the COVID-19 virus SARS-CoV-2. And it's at this point, after they've extracted the genetic material from the virus, prepared the master mix, where we've been to the preparation room, mixed the reagents, we then load what we call a 96-well plate into this machine, So this is just an example of the plate. We load this into the machine. Oopsie. We load it into the machine and it runs for about 1 hour. And at the end of the 1 hour, we're able to read from the samples we've tested who is positive and who is negative. We're then able to extract these results, share it with our data team, and our data team is made up of about, again, 11 individuals who work on a rota basis to collate the data from our case investigation forms received from the 6 counties, compile that data with the results that we obtain from these machines, and we then share that results with the county. I'd also just like to highlight this room behind me, what is usually used for malaria parasite culture. Again, it's a category 2 facility, and we've had to use this facility. We've expanded our work for COVID-19 testing in this facility when the number of tests increased. Following on from our real-time PCR tests, once we've identified the positive samples, we then hand them over to our sequencing team, and we've exploited our sequencing facility where we have a MinION nanopore and a MiSeq platform, and this allows us to look at the whole genome of the SARS-CoV-2 virus. Routinely in this lab, we would have been looking at malaria parasites. We'd have looked at viruses such as chikungunya or influenza or enteric viruses. And my colleagues here are preparing samples that have been identified as positive to look at the whole genome so that we can understand how is the virus moving through the Kenyan population, or how related is the SARS-CoV-2 virus to the global data set that's available on the online platforms.